Hello and welcome, ladies and gentlemen, for this second talk of the series pertaining to biohardware and the wonderful engineering possibilities which the protein molecule offers. And the beauty is that its structure can be encoded on a DNA. So, let me share my screen. So, the title of today's talk is Biohardware, the engineering possibilities with protein structure and macromolecular assemblies comprised of proteins. Now, this is the second talk of the series of 12, covering various aspects of engineering concepts applied to biological systems and to explore possibilities which may have been eclipsed due to domain boundaries. So, the biohardware possibilities with protein structure and macromolecular assemblies is the talk. Before this, of course, we spoke about cytoskeleton and the wonderful properties and functionalities of cytoskeleton. Now, to further it, we are adding the proteins to it today. Now, well, proteins, talking of protein structure, it is a long chain molecule, macromolecule, which is made of polypeptide bonds. So, between these beads that you see, there are polypeptide bonds, and the beads that you see are one of 20 amino radicals. So, totally there are 20 amino radicals. So, one of the 20 amino radicals is populated at each of the location, okay, at random as per the order specified on the DNA code as a recipe. So, this is the primary structure, and needless to say that the particular bead selected imparts specific properties locally to the chain. And apart from primary structure, which is a linear structure, as you can see, the property of polypeptide bond is that it leads to a rotation, axial rotation, and it has a tendency to form a spring-like helical molecule. Okay, so the secondary structure starts looking like a helical spring, and not only it looks like a helical spring, it also can function like one. Okay, like it can be compressed and it can go in tension, and depending on the relative strength of the beads that are there, it has a tendency to form elbows at certain point and bend. And when the length of the macromolecule becomes too much and it is getting extruded, so depending on the space available where it is forming, depending upon its birth cavity, so to say, it forms elbows and bends, and it takes certain shape which could be called as a globule. So it becomes the tertiary structure is like a globular structure. And further, the globules can have some hydrogen bond between them, and they could form a complex assembly, which is a quaternary structure, okay, which could have properties as specified by the specific beads at specific locations. Right now, it is also very much possible that these spring-like macromolecules could align parallelly, and they could have some cross linkages, and they could form pleated fan-folded sheets, or they can form a matrix or something like a fabric, something like a web. Okay, and the interspaces available could serve as capillaries. They could conduct some fluids or ions through intervening gaps. So that is also uh, a functionality which could be expected of these kind of protein molecules. Now, the globular structure that we spoke of, it goes and it has a tendency to get lodged in the membranes, various membranes, the membranes of cell membrane as well as membranes of vesicles and organelles, right? Now, the cell membrane is a liquid crystal kind of a thing. It is a liquid raft model where the inside is hydrophobic and outside is hydrophilic. Okay, so that it forms an impervious barrier between the media on either side, And of course, these anchorages, that are the globules, can provide some functionality like an open channel or a gated channel, or sometimes like a non-return valve also. Okay, so these are some of the interesting functionalities which the globules can impart wherever they get lodged into the membrane. And of course, they are electrostatically attracted to the middle portion so that they can't come out. And with the strength that they have and with the cytoskeleton that is attached to them, probably they can control the shape of the cell, and they can also haul the bigger vesicles. They can move the vesicles when the cytoskeleton has a tension and its length reduces. It can probably propel the vacuoles. Well, these are the 20 amino radicals, just for your reference. These taken, the disclaimer is that these are pictures are taken from public domain, and I claim no credit for them. And I must thank the content providers on internet for such an interesting content. But the idea of giving it here is just for you to refer that yes, these are the chemical structures of 20 amino radicals. These are their names. I'm not going into the names also. I'm not particularly interested in chemistry aspect of it. And these are the symbols which are used, 3-letter symbol or a single letter symbol when you represent a long chain of protein. Now, there are some groups in this one, like the violet ones are the ones which are electrically neutral. The green ones have dipole, that is, there is a plus and there is a minus. Okay, and the blue one has a net positive charge, and the golden yellow one has a net negative charge. So you have all the variety. Depending on which bead you select, you can impart a positive charge or a negative charge or a dipole or no charge. Okay, so that is one aspect. And of course, depending on the size of molecule and whatever atoms are there, it can impart different degree of stiffness and strength to the long chain that it is forming. Okay, so the bead could be acting just as a spacer in case it is a weaker radical. It could be just acting as a fill in the blank or a spacer in a long chain. So that could be imparting a weaker section to the long chain molecule. Otherwise, they could impart varying degree of strength, rigidity, reinforcement, which could be omnidirectional, or it could be in a particular direction. It could be imparting strength. So it could have some anisotropy by design, or it could be totally homogeneous isotropic properties. Okay, they could also result into tendency of bending and forming elbows wherever they are present in primary structure. So by default, the chain will start bending there. It will have tendency to bend. Conversely, suppose the chain is getting extruded, a protein is getting extruded, and it is encountering a barrier due to formative cavity that it is born into. So it will hit a wall and then it will be forced to bend, and soon that radical, whichever was there, will get replaced by the weaker radical so that it will become permanent. Okay, so that is another property which they could exhibit. And they could form chemical bond and hydrogen bond with the temporary quasi linkages, intermittent bond with the adjacent macromolecules, the vacillating bond kind of a thing. And they could form complex assemblies and complexes which are based on weakest possible interaction to be probed scientifically into further. Okay, they could have some residual electric charge, and of course they can respond to or create vacillating electrostatic field. So they can work like a transducer, or they could work like a motor. Okay, they can create a movement, or they can convert the movement into electricity, so to say, electric potential. They could also get anchored. We've spoken about it in the membranes, which have polar tails of lipid pointing inwards. So it could form a strong bond within. They could impart a tendency to oscillate because many of these beads or radicals have benzene kind of a structure. So there are jiggling bonds. So they have a tendency to cause jiggle wherever they are. So it is a lively macromolecule. It is not a dead and stationary molecule. It keeps jiggling to promote many possibilities of interactions, and many engineering possibilities come on account of jiggling. Okay. Now, with all this wonderful building block, what are the possible functions, engineering functions that we can envisage? One group is, of course, they can work as filaments and fibrils. They are extruded as a filament or a fibril, and they could work like a single strand or a fiber. They could work like a rope or a string for things like binding and bundling things together, for example, anchoring something with something. Yeah. It could be used as a haulage rope. It can pull the vacuoles, as I spoke before. It can pull some organelles. Okay. And it could navigate, and it could guide something that wants to find its way. Okay. So it could be like a rope carriage. Okay. So a guiding ropeway could be like a ropeway. It can navigate, it can carry things along a path. It can work like a flag mast for carrying at its head some signal, some message, some cryptic code, which it could pass to adjacent cells. Okay. So these are the various engineering possibilities that a fiber or filament or a linear member or a curved beam kind of a thing could be expected of. It could also work as a fiber that is extruded out of the cell, like keratin in hair cell, for example. So the long chain molecules can actually be extruded as a solid. And they can come out of the cell and have some other functionality as a tissue. Okay, so that's another possibility that it can manifest into. Then there are structural members which form the shape and structure of the cell. So these are could be relatively rigid structural columns, curved beams, flexible beams. Okay, mesh, web, flexible beam, flexible hoses, matrix, fabric, and so these are all structural things which can form the intrastructure of the cell. Then channels and tubules and capillaries for carrying fluids. So just like we find xylem and phloem kind of capillaries which are there in plant stem. So something like that, it's capable of forming. Of course, plant cells also come from there only, and also as structural columns, as a shaft that can rotate. And next category is, of course, they can function as movement effectors, something like a whiplash, something that can the sperm cells are known to navigate in a fluid, for example, through a whiplash. Yeah. So whip-like tail is there. Okay. A rope which can twist, intertwine, spool, and subsequently dissolve in cytosol, carrying out propulsion of vacuoles, and maintaining tension for doing that. So it's a functionality that we spoke of in the previous video also. Okay. So that one. And these ropes may also cause disposable fragments. The whatever parts, whatever components of cell get damaged. So actually, they can be pulled by these cytoskeletal fibers so that they drop into the cistern of lysozyme and they get digested there. They can get hydrolyzed there, and the contents can diffuse out through into cytosol through its membrane, to the membrane of lysozyme cistern. Okay, so that's the way the cell does scavenging function. Okay, vacuoles are bigger entities, so their lysosome goes and meets that vacuole, but otherwise scavenging can happen by some linear uh, haulage taking place within for the smaller macromolecules, for the fragments, for the fragmented organelles and stuff like that. and those could be dropped into the tank of lysozyme, which is very corrosive. It can dissolve everything, including the full cell also, if there is opportunity. There is evidence of one time motion actuators in plant kingdom. For example, there are carnivorous flowers in pitcher plant, which have a one-time closure of the lid. Okay, and then there are certain buds. If you touch it, suddenly changes shape violently and sprays the small seeds around. Okay, so there are evidences in plant kingdom of some movement causing elements which operate one time. Okay, then they could always work like spring. Owing to its secondary structure, it could work like a mechanical helical spring. It could work like a leaf spring or a suspension or a flexure. They could also form assembly of something like actin, myosin in muscle. They can form a micromuscle within a cell. So it's telescopic assembly of tubes and rods passing through them. Something analogous to hydraulic and pneumatic cylinders that we use in engineering for repeated reversible movements. The difference before and now is that this was a one-time actuation. This is a repeatable and reversible actuation. Okay, so micro muscle can also be there, and it's found in the cell. So, but these are actuated and deactuated electrochemically through some messenger molecules present in the cytosol. Depending on concentration of certain molecules, smaller molecules can get actuated and deactuated. Okay. Now these are what I referred some time back. This is a pitcher plant. Its lid closes when a insect sits somewhere here and it traps inside. It's a one-time actuation based on touch sensing. Okay. Muscle, on the other hand, right side is the muscle with actin, myosin, and these are the telescopically moving elements which move one inside the other. So this is the contracted condition and this is the relaxed condition. Okay. Certain molecules make it to contract, certain molecules make it relaxed. Okay, so it is a reversible movement causing action. Okay, and another category is, of course, they can work like drivetrain elements. So they can transmit force and energy like a lever or a shaft, for example, or even a molecular gear. Now, if you look at this picture, I mean, I'm not going into what this is, but then you can identify that this thing looks like very much like a molecular gear, this and this. And then these are the tubular columns, okay, which could act like a pump. And indeed, some arrows are shown as if they are conducting some things both ways. Okay, so they do carry out some kinesis of something as shaft, as spindle, as gear, as a pump. Okay, so all these engineering possibilities are also there, and inspired by these molecular level gears. Of course, engineers nowadays have started working on nanotubule based molecular gear, which could be used for mechanical transmission. Of course, they have nothing to do with the cell or organic part of it. They are non-organic, but it's inspired by this one, obviously, inspired by the nature. Yeah. Next range of applications that one can think of is they can work like a molecular level key. So they carry certain stereo specific shape and size, possibly with charge at some strategic locations carried by the globule, which is like a coded message or an ID tag, or could be even constituted as equivalent to a password. Okay, so a biological password at molecular level. How do you like it? So molecular lock is the one which is matching with the molecular key. So this is like a plug shape. This is like a socket shaped. Okay, so this is more like a glove shape. So whenever a key comes and fits perfectly into the glove with the matching counter charges at those locations, and the shape identifying the shape of the plug and the size, okay, and then something actuates on a perfect fit. Okay, so and it does further action. So molecular level key and molecular level lock are the functionalities which are carried out by protein structures. Then, of course, they can work as transducers and actuators, as I spoke before. because helical spring carrying a charge, uh, if there is electrostatic field, it will respond, or it will create its own electrostatic field if it is made to move. So both ways, it can work like motion sensor or like an actuator, so like a transducer or like a solenoid valve in engineering parlance. It can make something on or off. It can open or close certain ionic channels or valves on the cell membrane, and then the further functionality can be driven through that. Okay. Another interesting, lesser understood property of this one is as a modeling clay. In the cell, as much there are molecules, macromolecules, whose structure needs to be encoded on DNA, the spaces, the interspaces also need to be encoded. Okay, right? When you have a plan for a house, you do have to specify the empty spaces in the rooms, the carpet area, etc. Right? So based on that, the shape and size of the rooms, the shape and size of the birth cavities in which proteins are to be born, etc., have to be encoded and reproduced through the code or code of DNA. So nothing but protein is the media which can do flexibly the modeling and encoding of physical interspaces and their electrochemical surroundings within and outside the cell for the purpose of archiving on DNA. Okay. So you just assume that a new molecule is getting extruded, a linear molecule. It will go, it will build, it will go and hit some wall or some structure. Then it will be forced to bend. Then it will grow in a different direction and again hit something. Then again it will bend and go. Like that, it will form a globule whose envelope will represent the cavity that is there, right? So if the shape of globule represents the interspace, you can stretch it into a linear thing, read that code, convert it into nucleotide sequence, and put it on DNA so that the same can be reproduced wherever and whenever that context is there. Okay. So this is an interesting but lesser explored possibility, that use of proteins as a modeling clay. Okay. Then, of course, it can encode the space, but it can also form the interspaces. So it can form the mold within which the globules have to be cast when they are born. So it can replicate the encoded cellular interfaces and their electrochemical surrounding as per the archived document on DNA at specified mapped site at specific location. Genes are mapped specifically at certain locations on the cell. Okay, the macromolecules are not meant to just diffuse through the cytoplasm anywhere and everywhere. Okay, because they will get entangled within no time with so many things, right? So they have to be navigated, they have to be guided, they have to be shaped. Yeah. So next category is, of course, it can form something like a biocomposite material in line with CFRP and GFRB, carbon fiber reinforced plastic and glass fiber reinforced plastic, which have cloth reinforcement, and there is a resin. Okay, there is a solidified resin, and it forms the composite. So bone is an example of a biocomposite. Okay, so protein molecules create some sites in their matrix in a cloth-like structure, which get ossified, on which catch calcium, and then thereby they get some inorganic strength in otherwise organic and otherwise weaker structures. They suddenly get very solid and very strong, very hard structure. Okay, so binding calcium to form the bones, acquiring inorganic strength from the bonding of calcium to calcium. Okay, they acquire that kind of strength, and of course, macromolecular are also known to bind with organic dyes. I mean, that's how various colors are present in flowers, and dyes and substance like chlorophyll in leaves. Okay, so they can form a fabric which can bond with many kinds of molecules, many kinds of atoms, many kinds of pigments. Yeah, so to say. And Molecular level mechanically managing tools. This class is a shorter version of protein. They are called as polypeptide enzymes. These are summarily called as enzymes, and these are named after the process which they carry out. Okay, these are shorter molecular level tools which manage other macromolecules. Okay, as a vice, as a tool. Okay, they can twist, they can turn, they can join, they can splice, they can do all those actions on macromolecules. Okay, so these are called enzymes, and these are relatively shorter length protein, and they, since they are having energy and they apply force and move, they cause movement, they are also interacting with some sugar like molecules, ribose, etc., for energy and for carrying out movements. Okay, so that is the one. And of course, globular anchors, sensors, valve gates, we have spoken about. So that's the category. And last but not the least, in previous video, I have shown you some pictures of cell division and the spindles which carry out cell division, mitotic and meiotic spindle, which carry out cell division and which divide the contents of mother cell into two daughter cells, so that uh, chromosomes are split into halves, and one set of half chromosome goes in daughters, one daughter cell, another goes in other daughter cell, faithfully and repeatably, always. Right? That's another evidence that DNAs are mapped to cytoplasm, to specific location, to cytoskeleton. Okay, that's an evidence. Otherwise, it could not do that. Okay, so the interesting part is that the protein primary structure, only primary structure, can be encoded on DNA. That is the famous twisted ladder. Okay, and the code is formed of nucleotides: adenine, thymine, guanine, cytosine. So adenine and thymine combine invariably, and guanine and cytosine combine on the complementary strands which are there. And once it is unzipped and the strand having the real code is brought out through messenger RNA to endoplasmic reticulum wall, and there rRNA as an enzyme, as a vice, holds it, and it pulls it, it spools it, and the code is made to be read by a transfer RNA, and it forms at the other end a protein. Now, these are the nucleotides: adenine, thymine, guanine, cytosine. It's just an indication that these two shapes cannot exchange. A conical shape cannot fit a hemispherical cavity, of course. So, the pitch of 3 is used to encode one pitch of protein. So, each amino radical is represented by 3 pair of nucleotides. Okay, and depending on the code that they have. Now, this is the famous genetic code, again from public domain, so I don't claim any responsibility or credit for this one. But I'll just explain so that you can refer it if you have to. Like in mRNA, T, that is thymine, is replaced by uracil. That is one change. So you don't see T here, instead you see U. But if you want to read, you have to read it radially outwards. So U, C, and one of these, they all will represent the amino radical serine, for example. So it is having redundancy, obviously. So many codes represent the same thing. But vice versa is not true. Same code cannot represent more than one amino radical. So there it is unique. And apart from 20 amino radicals which you see in this wheel, there are some codes which are reserved for start and stop, because there could be a framing error. Suppose you start after one pitch, the entire meaning of genetic code will change. The entire sequence will change. It has to be read in correct direction and starting at correct point. Okay. Otherwise, it would mean something else totally. Right. So, yes, there is a start and stop marker on the messenger RNA, which is implemented. Okay. So, mRNA is the one which reads the code sequence, which reads the gene through DNA, which has been unzipped. So, it takes a copy, it makes a transcript, and then it carries the transcript to the wall, to the rRNA, that is ribosomal RNA, which has some energy. It works like a molecular vice. It's able to thread and pull and unspool the mRNA towards it, and it presents the code of 3 in groups of 3 to tRNA head, and tRNA is the transfer RNA, okay, which looks something like this. So this is a tRNA for reference. So it has got some loop, T loop and D loop, etc., like 2 years. So the 2 years are held and it's oriented so that this anticodon segment is juxtaposed with the codon segment of mRNA. So these are the counter codes which match with the code of this one. They are complementary actually. So they can bind with the 3, so to say. So the pairs will bind. When the 3 pairs bind, it produces some signal, and then whatever amino radical was carried by the tail of this one, it gets incorporated into the chain which is getting extruded, the protein chain. Okay, so this is a new member in that one, new bead. Okay, after that, this thing gets discarded, and next codon comes, and next tRNA comes, and that cycle keeps on going until the protein gets formed, until you encounter stop code here. Okay, so that forms a complete protein structure, primary structure. Okay, now the interesting part is what makes sure in cytoplasm that corresponding to this anticodon, only certain amino radical is present here, and what is preventing a mix up or mistake there. Okay, but mistakes are not known to happen. Genetic code is universal. All type of cell, all type of organisms. So it is kind of universal, and it has evolved to be universal that way. The family of tRNAs must have evolved. Okay, so the suppose this tRNA loses its amino radical. The next job for the cytoplasm is to populate this again with the same another amino radical, the same one, the same bead should come here. Now that molecule which does that is named interestingly as amino tRNA synthetase enzyme. Now what can you make out of it? It's just a function. We don't know how it does it, but surely it does it. Something does it. Okay. So that is the mechanism that is responsible for universality of genetic code and foolproofing of genetic code. So it must be some short protein-like molecule, some enzyme, which holds tRNA by the loops in a vice kind of fixture, so that anticodon gets juxtaposed to the codon. That is one function. Other function is upon mismatch. Suppose the codon does not match the anticodon, that tRNA should get thrown off, and next one should be picked up. Okay. Upon matching tRNA, it aligns with the protein being synthesized and dispenses the carried amino radical in the primary sequence. Okay. But the tricky part is how it is ensured that proper amino radical gets synthesized with that tRNA and gets replenished again according to the anticodon carried by it. And there is never a mix-up. Well, surely, probably it is the specific shape and field of anticodons which is somehow matched with the complementary shape and field of amino radical. So that is the only engineering possibility that it could be like that. Considering the universality of genetic code, it's still a mystery from where these molecules that make above happen, from where these molecules come, who makes these macromolecules, and what about their own genetic code? How do they get formed? How do they get stored on DNA? How they are formed in the first place? Okay, so it's a chicken and egg situation. tRNA is responsible for the translation of code, but what is forming tRNA itself? Okay, somewhere it has to start. Now, it has never been possible to synthesize a cell that does not descend from another pre-existing cell. Okay, with just nucleus, with just DNA, you can't make a cell, you can't make an organism. It is as much the genetic code as the seeding cytoplasm. So it requires a seeding cell. It requires the environment of a cell, cytoplasm, cytoskeleton, membranes, organelles present therein, which forms the apparatus which together complete the prerequisites for making a new cell, making a new protein. Okay, so that's all for the day, and see you for the next talk. And the next talk will be uh, dealing with the genetic archive. How does it work, and what are the unexplored mysteries and evolution of genetic archive? So with this one, we can conclude for the day, and do see the next talk as well. Bye bye.